Hey everyone! In this video, we'll examine the life cycle of eukaryotic cells called the cell cycle. After you've watched this video, remember to fill out the learning guide. And by the end, you should be able to answer 2 questions.
And our 2 questions are, what events occur in each part of the cell cycle? And how do these events lead to the production of 2 cells from 1 cell? The cell cycle of a cell is the process a cell goes through to replicate itself. As we saw in Module 1 of this chapter, cell replication is important for the development of an organism and for the replacement of cells. In order for a cell to replicate itself, it must grow, replicate the chromosomes, and physically split the cell. The cell cycle is a cycle and not a linear pathway because cells can go through it over and over, producing many cells over time. The cell cycle has 2 main parts. The first is interphase. The prefix inter means between. Here, interphase is the time between cell divisions. M-phase, or mitotic phase, is when the cell divides to form 2 daughter cells. Interphase is split into 3 parts: gap 1 or G1, S-phase, and gap 2 or G2. M-phase has 2 parts: mitosis and cytokinesis. During G1, the cell grows and replicates its organelles. This ensures that once this cell divides, the 2 daughter cells will have enough cytoplasm and organelles to function. And it ensures that the cells won't get smaller after each round of cell division. However, some cells do divide without growing, and the daughter cells do get smaller, as we'll see later. During G1, the cell also makes enzymes and molecules necessary for the replication of chromosomes, which will happen in the next phase. Lastly, the cell carries out its function. For example, the function of this cardiomyocyte, or heart muscle cell, contracts in rhythm. It beats. Many of these cells beating in unison allow the heart to expand and contract, pumping blood through the body. Cell functioning and growth can continue throughout G1, S, and G2. Not all cells move through the cell cycle constantly. Some cells may exit the cell cycle at the G1 phase and enter G0, a non-dividing state. If subjected to the right growth factors and cellular environment, some cell types can re-enter G1 and proceed once again through the cell cycle. Other cell types seem to stay in G0 for the rest of their existence and undergo extensive structural changes. These cells are called terminally differentiated cells. However, whether certain cells can divide again within the human body is difficult to determine, and some scientists argue that even very differentiated and specialized cells may divide again under certain conditions. After G1, the cell enters S phase, named for the synthesis of DNA. This is when the chromosomes are replicated. At this point, cohesin holds the sister chromatids together. Next is G2, where the cell continues to grow and produces molecules and proteins needed for cell division, which is next. This includes the tubulin proteins, which make up the microtubules or spindle fibers needed for chromosome movement. G2 is also a time where the cell detects any double-stranded breaks in the newly replicated DNA and repairs them. These breaks can be caused by exposure to irradiation, ultraviolet light, or other mutagenic agents. Double-stranded break repair can occur in S phase as well because one sister chromatid can be used as a template to repair the other. Because, sisters help each other. After the completion of interphase, the cell enters M phase. M phase has two parts, mitosis and cytokinesis. During mitosis, the chromosomes condense and are separated into two separate nuclei in such a way that each nuclei gets two complete sets of chromosomes. Mitosis itself is divided into several phases, which we'll examine in the video on mitosis. During cytokinesis, the cell actually splits, forming two daughter cells. Each cell gets one nucleus and two complete sets of chromosomes, which is what the parent cell in G1 started with. Cytokinesis is quite different in plants and animals. In animals, a series of long fibers forms a structure called the contractile ring around the edge of the middle of the cell and pulls the cell membrane inward. This creates an indentation in the cell membrane called the cleavage furrow. Plant cell membranes can't be pulled inward easily because they're surrounded by a rigid cell wall, and also because the plant cell membrane is under positive pressure from all the water inside the cell. So plant cells separate the two cells by building cell membrane and cell wall from the inside of the cell outward. This new structure is called the cell plate and will eventually merge with the existing cell membrane and cell wall, separating the two cells. Here's a super cool time-lapse video of a cell going through mitosis and cytokinesis. The DNA is labeled red and the spindle fibers are labeled green. Although the diagram we've drawn on the last page made it seem like each phase takes about the same amount of time, this is not the case. Human cells in culture can complete the cell cycle in about 20 hours, with most of the time spent in the G1 and S phases. However, how long a human cell takes to complete the cell cycle and how long it spends in each phase can vary among cell types. Yeast undergoing fission can take about 2 hours to go through the cell cycle, with most of the time spent in G2. Cells in a Drosophila embryo complete the cell cycle in an amazingly short amount of time, only 8 minutes. Early embryos are just zygotes that have begun to divide. These Drosophila cells basically skip G1 and G2 and divide without increasing the size of their cells, so the cells do get smaller after each division, at least for the first several divisions. We can see the same thing happening to these xenopus or frog embryos. They divide without increasing the size of their cells. Let's see it a few more times. Now that we've seen what happens at each phase, let's follow what happens to the number of chromosomes, DNA molecules, and gene copies as we proceed through the cell cycle. We will use this fictional 2n equals 6 cell to demonstrate, and we'll follow the A, B, C, and D genes on these chromosomes. Here is our G1 cell, with 2 of each gene and chromosome type, 6 total chromosomes, and 6 DNA molecules. When the cell goes through S phase, each chromosome is replicated. So the number of DNA molecules doubles to 12. Since we now have 4 copies of each chromosome type, we have 4 copies of each gene. The new copy of each chromosome remains attached to the original at the centromere, and since the 2 sister chromatids are attached, they form a single chromosome. So there are still 6 chromosomes in this cell at the end of S phase. The same is true for the cell at the end of G2. We'll go through how the chromosomes change during mitosis in the mitosis video, but by the end of mitosis, the chromatids have been pulled apart and sorted into 2 nuclei. However, the cell has not yet split, so there are still 12 DNA molecules in this single cell. Since they are unpaired, there are also 12 chromosomes in this cell. And there are still 4 copies of each gene. By the end of cytokinesis, the cell has split. So the daughter cells each now have 6 chromosomes, 6 DNA molecules, and 2 copies of each gene, which is what the original G1 cell started with. This is the whole point of the cell cycle, and all the things that occur within each phase, to produce 2 daughter cells from 1 parent cell.
Okay, so at this point you should be able to explain what occurs at each phase of the cell cycle and how these events lead to the production of 2 cells. If you can't, as always, feel free to watch part or all of the video again, and remember to fill out the learning guide as well. Now in the next video, we'll take a look at mitosis. See you then.